Student Dr. Umar came to KCU with a BS in biology from the University of Texas at Dallas, where he graduated cum laude and gained experience as an undergraduate researcher. He worked in the pain and neurobiology lab for 2 years under Dr. Michael Burton and Dr. Theodore Price on projects aimed at understanding the role of aging, sex, and neuroimmune interaction in the development of neuropathic pain. He is co-first author on the paper Integrated Optical Clearing and Twophoton Imaging to investigate sex differences in neuroimmune interactions after peripheral nerve injury. He is currently a third-year medical student at Kansas City University, and since joining, he has served as president of the Internal Medicine Club and volunteered as a health coach for children at increased risk for obesity. After obtaining his DO from KCU, he's interested in pursuing a career in anesthesia and pain management. Umar, I will throw it over to you. Remind everyone, please mute your mics. And if you have any questions, just raise your hands or shout at the end and we'll be happy to take them. Thank you.
Awesome. Thank you for that introduction. Let me go ahead and share my screen and get the PowerPoint put up. Today, I'm presenting the research that I've done over the last couple of years and worked in the summer of first year. What we're talking about today, as the title says, is the use of optical clearing to investigate sex differences in pain after like peripheral nerve injury. And so I know that's a lot of complicated terms. So this presentation is going to serve to go through sort of everything in the title, and we're kind of going to break it down, and I want to explain it in a sort of comprehensive manner that we can all explain. So first, I want to start off with the basics. So why are we doing this research and why do we care? So a lot of us are clinicians over here, or at least know someone who suffers from chronic pain. We know that it's a pretty big problem. You see it in the media all the time in regards to the increasing prevalence of pain, as well as all the side effects that are associated with it, whether it's regards to the increased opiate use, the whole opiate epidemic going on, as well as the fact that there are very limited treatment options for chronic pain, because a lot of people sometimes assume that opiates are sort of the answer, that they might be addictive, but at the same time that they provide significant relief. However, like it's found, especially with chronic pain, that's not really the case, and that they just lose effectivity and they don't really work, especially for nerve type pain. And so when you think nerve pain, people think of things like sciatica, that's the most common one, and other common nerve disorders, but which usually people ascribe to being happening after an injury, an acute injury, for example. But a lot of the times, it'll be things like post-surgical pain, where like people undergo routine surgeries, and they suffer from chronic pain afterwards, chronic nerve pain, whether a nerve is damaged. There's a multitude of reasons. And so it's not just more like the specific examples that we think of there, it's a lot more prevalent than a lot of people might think in regards to how much of this is going on. And an important thing to note is, while that is quite a horrible aspect of all of this troublesome stuff going on, there is even more to that, where like the sequelae and the long-term effects in regards to increased comorbidities of depression, sleep disturbances, and a multitude of other things that come along with whether it's using opiates, whether it's suffering from chronic pain. So that all combines and adds to this effect. And some estimates are that it has an economic impact of over $635 billion a year. And the people that did that didn't include people under 18 or people that are institutionalized. So it's likely higher. So it's quite a significant burden. It's one of the largest reasons why people miss work. And so economically speaking, quality of life speaking, and in terms of patient care, it's becoming even more of a significant issue than it was before. And as I was saying earlier, the poor management of this, whether it's opiates or nerve agents like Lyrica or gabapentin, a lot of times don't have much effectivity, and they can lead to other things like dependence, addiction, and the side effects that come along with just those medications. And so the one thing to note is when I talked a little bit about just the overarching view, the current literature shows that males and females don't really perceive pain in the same way, that there's a clinically significant difference between males and females. And with a lot of previous research, previous funding, that's something that wasn't really, people didn't really look at that, or people didn't really dive into it. And so that was one of the main focuses, or the main focus of our specific project, was trying to elucidate the differences between males and females, because that's something that's starting to pick up a little steam right now, but it's something that we don't have many answers on. And it can serve to sort of get more catered therapeutics, or to even clinically just approach males versus females differently, if we know why the reason is, why females feel a pain in a different way, or why they report more pain, and why they suffer from more pain. And so all of these problems combined make it a very important paper, is why we wanted to go look into it. So I want to go ahead the next couple of slides. I want to talk a little bit about the main component of what we looked at in this paper. And so one second, there we go. So we went ahead and looked at the neuro-immune interactions that lead to chronic pain and the sex differences between those. And so to go ahead and simplify that, we went and looked at macrophages, which are particularly known based on the literature to play like a significant role in chronic pain. And we'll go ahead and dive into the nitty-gritty details in a little bit. So the macrophages are involved, and macrophages, like the state that they're in, whether it's a certain classically activated or a alternatively activated, the state that they're in helps play a significant role in sensitizing neurons in a particular manner. And so, for example, right here, so the two different sort of shapes that we're looking at is, like, on the left-hand side, it is elongated, or the fancy word for that is prolate shape, and on the right is flattened and oblate shape. And in terms of like an immunological standpoint, these correlate with like alternatively activated, also known more as like the helper, like not the helper, but it's the one that basically helps decrease inflammation, like more repair remodeling processes, and the classically activated M1s, which is what we think of as inflammatory and like sort of the bad, to put it colloquially. And so from the literature showing that there are associations between macrophage morphology, or another word for that is shape, as well as what kind of activation states they're in. And we already know in terms of what activation states mean in regards to like what cytokines and inflammatory markers are released. We like aim to further elucidate whether there are any sex differences in relation to nerve injury in this experiment that we did. And so a couple of points to note about the experiment. Overall, like, things in the lab aren't going to be the same as in real life. And so it's an important point to take note of. And as I was saying earlier, not much research comparing both sexes directly, it's a severe lack that we need to address. And sometimes other literature has shown, people have sort of compared slightly in terms of some differences or whatnot, but a direct head-on-head comparison is there aren't as many of those. And then another aspect that we looked at is we used full whole tissue. And so a lot, traditionally, people would slice tissue and go ahead and run immunohistochemistry on that. And so instead of that, we used a whole 3D tissue. And so I'll go ahead and further elaborate on the whole process and everything in a couple of slides. But one of the novel aspects, or one of the more thorough aspects of our experiment, was the two-photon microscopy that we used. So if you're not too familiar with imaging, just to provide like a little bit of a background, traditionally you have your normal light microscopes, and the sort of best, the better imaging modalities that are available right now are confocal microscopy and two-photon microscopy. And the primary differences between those is, if you see on the top diagram right over here, this talks about the excitation and emission wavelengths of how like a fluorescently tagged cell or molecule works. And so on the left-hand side, or on the left side of this whole diagram, this shows a more traditional confocal microscopy, which excites with just one photon, and that's how you reach your emission fluorescence. And it goes ahead and kind of like stimulates the tissue sample. And in order to focus the image, there's a pinhole right in front of the detector that sort of filters out essentially the other background light. And that's how you get a more focused image. So the downside to that is that even though this is filtering out the other sort of extra fluorescence that's coming from up top over here, the other tissues are still getting stimulated with that photon, and it's not very specific. On the other hand, two-photon microscopy is sort of like the most specific out right now in regards to imaging. And what it does, it uses two photons at an infrared spectrum. So this is quite a bit higher energy, and it uses these through quick pulses because it requires quite a lot of high energy. And what it does is it releases like one particular photon. So it's very, very specific in regards to like just a general area of the sample versus one specific point. And so this prevents, so like I was saying earlier, it uses the near infrared excitation, which prevents the tissue prevents the light that you're using to stimulate the tissue from kind of going everywhere and scattering and being more specific. And we don't need to worry about the pinhole like we did with the confocal microscopy because there's just a lot of less background noise. And a big thing which helped us in our experiment was that it goes further into the tissue, while a normal confocal microscopy could go only at the surface level. Let's say for a full brain, this can go quite a bit deeper at like a couple millimeters almost. And so it allows you to get a whole picture image of what you're trying to sample, as opposed to sort of slicing it and doing it and kind of putting it together using computer programming afterwards, computer software. And then reduced floater breaching. If you go ahead and stimulate these fluorophores or these molecules, essentially, they can only do it so much. So after a while, they just don't work anymore. So essentially, since it's so specific and it's not stimulating things it doesn't want to be stimulated, they don't stop working as early, essentially, is what that means. And so on the bottom over here on the right, this is a paper that was released by the lab where they went ahead and they did in vivo two-photon imaging of mice in the skin. And so if anyone's interested, that'll be available to go check out. It's a video that was published on Jove. All right. And so now this is more of personally, I think the part that's always nice to look at is the tissue clearing portion of the experiment. So recently there's been a lot more of interest in tissue clearing, which essentially is exactly what it sounds like. It's taking a tissue and clearing it to the best of your ability, and with the goal of having it easier to image with a multitude of other benefits to it. And so while there are a lot of other methods on how to go about this, I'm going to talk about why we chose scale, which is the tissue clearing method that we used, and the sort of general process in regards to tissue clearing. And so a couple of benefits of this: it preserves tissue integrity a little bit better than some of the other methods, preserves the fluorescent labels, and so it doesn't interfere with the whole emissions excitation. It doesn't sort of add any reagents that might interfere with that. So that was a benefit. And one larger problem that's usually common in clearing protocols is that the tissue, in regards to how it works, will kind of expand the tissue and make it swell up. And so this had less tissue expansion as compared to other things. And so overall, on the bottom over here, this is a general overview of how scale works. So the reason why cells cells are sort of opaque and have when tissue has an opaque color is because of like the lipids in like the cell membrane. And that's the reason why they look how they look. And so the general principle of what scale does is it removes the lipids from the cell membrane, allowing the tissue to be essentially clear. And so the primary thing that it does it by is urea, and you add a little bit of a detergent, usually like Triton is what they use, or what we used as well. And so you go ahead and that sort of opens up and helps basically osmotically almost drag out the lipids out of the cell membrane. This is about a 10 to 14-day process what we used, but people go ahead and put it in the solution for about 2 to 3 weeks. And it's always a little bit of fine tuning depending on what tissue you're trying to clear, as well as how well the tissue is doing itself. So this is a process. While the protocol is there for how long to do it, it's a little bit of a trial and error in regards to, you don't want to have the tissue expand too much, and have the solution be too aggressive, because that would make it harder to image, or it just kind of almost dissipates away. But at the same time, you can't wait like months and months for tissue to clear. But overall, that's the general process, and this is what we used in our experiment. And so what this does in regards to imaging is it brings the tissue closer to the refractive index of what you're trying to image. And so the refractive index of water is about 1.33, and scale is about the same at 1.38. And so if, as you can tell on the top right, if you, this is a little bit more of like a physics sort of explanation around it, but if you can go ahead and equalize the two refractive index from like in the tissue sample to like the solution medium that you're using to image it, that provides a more clear image. It allows you to penetrate the sample that you're using. It allows you to go deeper, and essentially you're able to get a better image. And this is a diagram of sort of the body composition. And essentially, as you can tell on the bottom, as I was saying earlier, the lipids is what we are attempting, or what we aim to remove, and how the process of scale works. And so, as I was saying earlier, the clearing is used to sort of get the refractive index equal amongst both the tissue and the sort of solution medium, thereby providing like a sort of even road for like the lasers and the emission and all the photons to travel on, instead of having it like sort of like this arrow going like all the way one way. It would kind of sort of be a little bit more equal in regards to like the light path and how it travels. And so those were a couple of that was like some of the main important points in regards to the different, like, I guess, novel aspects of our experiment. Now, so what we were looking at was macrophages and how they are different in regards to males versus females in clear tissue. And so we had a couple of hypotheses. So we hypothesized that there was going to be an elevated leukocyte recruitment to peripheral nervous tissues following nerve injury in males. And the main leukocyte we were focusing on was macrophages. And then the second one we had was, we hypothesized that there were going to be sex-dependent differences in regards to morphology, which was like the shape sort of characteristics and qualities after SNI. Now I'll go ahead and talk about what SNI is. It's a model of nerve injury. And so I'll go ahead and I'll bring up the diagram that go ahead and talks about. So over here, you can see a couple of our images and our findings that we had, but I want you to go ahead and focus your direction up here towards the top. This is like an overview diagram of our entire experimental procedure. And so, this initial portion with the mice over here, that sort of signifies the sort of injury model we use. So SNI, also known as spared nerve injury, is a process where you go ahead and you take the sciatic nerve, you go through surgery, you go ahead and find the sciatic nerve, and you ligate it has 3 branches. And so you ligate 2 of the nerves and leave 1 intact. And this is like it's a long proven method in terms of inducing neuropathic pain in an experimental animal model. So we would go ahead and surgerize the animal, go ahead and find the sciatic nerve as well as its 3 branches. Like I said, ligate 2 of the nerves and leave 1 of the nerves intact, and wait about 3 to 5 days and behaviorally test the animals throughout this time period to ensure like they have chronic pain, and their paw withdrawal thresholds in regards to filament testing would be they'd be more sensitive to it. And so we go ahead and perform this surgery. as well as like a sham surgery. And so sham is our like control essentially. And so what that entails is that you go ahead and you kind of almost pretend like you're doing the surgery. You go ahead and dig through the tissue, you find the sciatic nerve, however, you don't cut it. And so that's to ensure that like any localized inflammation or any to control for anything that might be like any local inflammation just from the surgery, and to ensure it's more specific to just the nerve pain that we're trying to induce in this animal. And so the sham animals did not have any of their portions of their sciatic nerve, any branches of the sciatic nerve cut. So after we've induced the nerve pain in these animals, we went ahead and harvested tissues from the lumbar DRGs, or dorsal root ganglia, which is like a primary anatomical component, nerve component that's involved in pain. And we harvested the entire sciatic nerves. And we used lumbar because that's the main areas where the sciatic nerve is innervated from. And the sciatic nerve, we wanted to see if there was any changes in that as well. Put that into the scale solution on like a tissue rotator for about 10 to 14 days until like they sort of cleared out. These were smaller tissues, so it was definitely a little bit easier than say doing a whole brain compared to like, you know, something like these nerves, and these nerves are quite a bit smaller. After that, went ahead and did our 2-photon microscopy on these and analyzing, following analysis with Umar software, which helped us quantify the findings that we had. Because I mean, while these images are quite pretty and nice to look at, we wanted to have like a concrete, you know, objective way to go ahead and quantify these findings. So over here we have images of the dorsal root ganglia. So sham, as I was saying earlier, is like a control group that didn't have their nerves cut. So IPSI would be the same side as like where we did the pretend surgery, where we did the control surgery, and then the contra would be the opposite. And then the SNI would be the spared nerve injury surgery. And then same thing, same IPSI, same side, and contra the opposite side. And so this kind of gives you an idea of sort of what we looked at in terms of when we found the images. And I'll be presenting the data, like the information in just a second. And so as you can tell, like post nerve injury, there's definitely an increase of leukocyte recruitment, or macrophages essentially, that are genetically tagged in these animals. And so going on to the findings, over here on the y-axis of the graph, you can see lysozyme M TDT positive. And so we used a very specific cell line, because I kept talking about what we were looking at macrophages, but how did we look at just macrophages? So what we did is we took a specific reporter line where only lysozyme positive cells would be fluoresce red essentially. And so in the lysozyme can be expressed in like other immune cells, but in the peripheral nervous system is what we were looking at. Macrophages through the literature have been shown to be the primary cell responsible and primary cell that responds to nerve injury. And so that kind of helps us know, okay, it's not going to be other lysozyme positive cells, because in the setting that we're looking at, other cells don't infiltrate. It's macrophages that are the primary player. And so we had significant findings in the ipsilateral male side of lysozyme positive cells, where they had an increase in their DRGs. And so as you can tell over here, we had male ipsilateral, male contralateral, and then same with the females. And so, yeah, for sure, males on the ipsilateral side had higher lysozyme positive cell recruitment. And over here, we went ahead and did a ratio to go ahead and compare ipsilateral versus contralateral side. And so we went ahead and noticed the same thing over there. Males on their ipsilateral side, compared to like their sham or contra side, essentially did have a significantly higher amount of these macrophages essentially. So that goes to show that yes, there is a difference and there's an increase in males, but we didn't notice any significant difference. So it helps sort of give us an idea of that there is something different going on between males and females, and sort of further, like there's no one knows the exact cause right now, or the exact physiology that's behind that, but that's, you know, something for further research. So go ahead, I'm gonna go ahead and talk about a little bit about the morphology aspect of our experiment now. And so, as I was saying earlier, oblate or flattened corresponds to M1 or classically activated macrophages, and then prolate and or also another word for that would be like an elongated shape, correlates more to M2 macrophages, which are more like the anti-inflammatory, and prolate you can think of as, flattened you can think of as more as an inflammatory classic macrophages. So in going ahead and getting the ratio for that, we didn't find anything significant in regards to this portion. However, we noticed just some trends to, I guess, take into account or look at maybe topics of further research, where female sham IPSCs had a higher proportion of the flattened or the M1 type macrophages, which for the sham, but for the IPSC, it was sort of a little bit lower, and they had more of that M2 elongated shape. So like I said, no significant findings, but sort of some more for future questions that could be posed from this, like, if, doing it with like a slightly different model, like what potential, findings could there be, and what would that mean in regards to future directions for other research to perform. And so this slide is, it gives a pictorial example of, or a pictorial graphic of what I was talking about in regards to the nerve injury. So as I was saying earlier, this is the sciatic nerve over here, and we'd go ahead, and this red mark over here signifies that we're cutting two of the nerves, and we're leaving the sural branch, one of the nerves, intact, therefore emulating chronic nerve pain. And so there's like a molecular sort of crosstalk where the immune system and nociceptors, or the pain receptors, go ahead and kind of talk to each other and sort of get sensitized, and the macrophages are able to sort of sensitize one another. And so overall, a nerve injury leads to the sort of talking in between, this molecular crosstalk in between these two. Chemokines, cytokines, DAMPs, these are things like, IL-6, a lot of the stuff that we've learned personally, in our first couple of years in regards to the inflammatory effects of this like injury. And they're also implicated in many other conditions like asthma and just like acute injury as well. These aspects get upregulated, and that leads to, an activation and sensitization of nociceptors. And so macrophage will become activated, release cytokines, chemokines, and DAMPs, which are damage-associated molecular proteins. All signs are involved in like injury processes in the body, and this eventually leads to peripheral neuropathy. Now over here, up top, this colorful diagram is signified males and females. So this would be like a cellular view of the DRGs, or the dorsal root ganglia. And so this is a pictorial example. So our spine would be, or our spinal cord would be right over here. These would be the nerves coming out, and these kind of form to go ahead and lead to the sciatic nerve, which goes and further branches out. And so if we go and look in those pictures of the red dots that we saw earlier, this is a graphical version of that. So in males, these sort of purple cells are like the lysozyme deposit, the macrophages that we were discussing earlier. There's an increased cell infiltration, increased volume, and increased ferocity in males, which was not seen in females. And then when we went ahead and did the same imaging and the same process of looking at these macrophages in the sciatic nerve, we didn't notice any significant difference, as I was saying earlier, but we also went ahead and took a look at proximal versus distal in regards to if there might be any trafficking differences. But like I said, there were a slightly increased cell infiltration in males, but we did not find anything significant. But we also looked at morphology and did ratios for everything. And so some future directions in regards to our experiment. So as I was saying earlier, pain, it's becoming a bigger and bigger problem in America. And so the therapeutics, understanding this process, all sort of begin with the basic sciences and understanding how this works, what this molecular crosstalk between macrophages talking to neurons, and why there's a sex difference in between these two. Understanding those processes will help cater our like research further along into like what we should be looking at, as well as would provide a better target for therapeutics. So whether it's drug targets or physical surgeries, it would help guide us in terms of females versus males. Do we have a different clinical treatment approach in regards to treating pain in people? And then, as I was saying earlier, one of the most important things, at least for a lot of clinicians, is how do we help our patients? And so, applying this information in a clinical setting is key. And so, while a lot of times this can be a little bit seem a little further along, it's an issue that needs to be addressed. And so, with the future goal of being able to have a protocol or have an idea of approaching a male versus a female patient different, because it's something that we're going to deal with and face on an everyday basis. And so having more guidance on that and more of an understanding is something that's very beneficial. And so I want to go ahead and thank like Dr. Michael Burton, that who's my PI that I work with, and then Thomas Party, who's one of the grad students there as well, who definitely helped me out a lot in terms of learning this whole process and throughout like my lab process. And then all of my teachers, a lot of who are here right now, who've taught me throughout the first couple of years and helped me get a solid foundation, as well as everyone who came here. So thanks everyone for coming. And then that is it for me. So if anyone has any questions, I'll be more than happy to answer.
Thank you, Umar. That was a great talk. I was kind of curious. I had a question, so I'm going to start. When it comes to the macrophage part, can't you collect macrophage from blood samples? And if you can, could you collect from humans and look at the same morphological differences in humans at different times that they would be in pain, male versus female? So I'm thinking like we both get kidney stones, and those are very painful. Could you compare, what's happening in a male at the time of their kidney stone versus the time of, a female, and start to make those same comparisons in humans?
So no, that's a really good question. So that's something I guess I should have discussed a little further. So while like for an acute injury like that, for kidney stones, you might be able to do that. While I'm not fully sure, with chronic pain and chronic nerve pain, like peripheral nerve injury, which is sort of thing the topic of focus, those don't. That's sort of like a separate ecosystem in a way, where like the macrophages won't, like there are macrophages in the blood, but those are different in regards to they don't cross over there. Sometimes there's literature that says some of them cross over and go into the peripheral nervous system, but it's a more localized process. So you can only be able to look at, in the nerves per se, and in DRGs, and that's why we'd have to harvest those and look at those. So while that is a pretty good idea, and there's people that are trying to look into things like that, the main thing right now is that there's not enough of them moving over to go ahead and quantify that. And like the main differences in upregulation are more of like localized inside the peripheral nervous system, with like the whole crosstalk going on with the nociception.
Okay.
But did that answer the question?
Yeah, that makes total sense. It was very enlightening for me. So thank you very much. I appreciate that. We have a couple hands up. Dr. Johnston, go ahead, please.
I think Dr. Wolf was ahead of me.
Did he? Okay. Dr. Wolf, please go ahead.
Sorry, I was going to say I enjoyed your talk very much. Your explanation of the two-photon stuff was, I'd seen two-photon stuff before, and you really did that justice. I mean, I think I finally understand it a little bit more this time, but I was curious going into it of how are they measuring the macrophages? And so you finally gave me, provided that they were traced macrophages. So that was helpful. But so I enjoyed your talk a lot. I was curious why you were focusing in exclusively on the dorsal root ganglia. I mean, I'd also be interested in like the substantia gelatinosa region or in the peripheral tissues, and to the extent that you could have maybe extracted some information out of there. And then as long as I'll shut up after this one, I was also curious if you looked at the different cytokine levels. I mean, was there Th17 or various other things that were more expressed in one sex versus the other, one gender versus the other?
Those are all great questions. So let me go ahead and let me know if I'm going to miss any of them. Let me go ahead and address each one of them. So with the cytokines, that's something that we weren't specifically looking at in this experiment. It's just beyond in regards to like the scope and what the reach we were going for. But that is a question that we've brought up. There are literatures studies right now that are quantifying a few different cytokines in regards to the upregulation in males versus females, and in regards to after like an injury, but we didn't specifically look at that. Now, in regards to using the dorsal root ganglia as opposed to acute tissues, while there are changes, yes, in like the local injury from when we do the surgery and from other tissues in the body, the sort of the chronic pain that happens in the nerves, essentially. If that's a colloquial way of putting it, but yeah, so the long-term process of like sensitization and because they won't have any if you sort of wait a while and wait for the injury to heal, after all the acute injury is healed, they won't have any sort of localized inflammation in the area, and any other like acute sort of inflammation, but they will still be hypersensitized due to like the interactions that are happening primarily in the DRG, which is where primarily the nociceptive fibers and the cell bodies of the nociceptive pain sensing fibers are live. And so that's the primary reason, and that's through quite a bit of literature and pain research, but that's the primary reason why we went ahead and focused on the DRG, because they're like they're like the main players responsible in the perception and the transition to chronic pain and the persistence of chronic pain as well.
And so those changes that you saw in the macrophages persist. I mean, that was among the things that I was interested in, was after, I mean, they persist throughout chronic pain, or do they wax and wane? And because I was curious whether or not what you were looking at was just something that you happened to see at the instant that you happened to look at versus chronic pain, which lasts for years.
That's a great topic that you bring up. And so with this one, I mean, we did look at specific time point in this one, but overall, if there are like other studies that the lab has done itself, where you go ahead and we did an experiment, for example, I helped work on with aged rats, but once the surgery is performed, even like a year after, these behaviorally, at least, these mice would be hypersensitized, and like I said, everything is healed, like the surgery is healed, but this behavior persists. And there is also data out there that like these changes as well exist. There's upregulation of other, like I said, damps and things like that, but there's upregulation of or just sort of overall change in regards to long term. So these are persistent, if that answers the question, and they do last even when the acute changes are overcome.
Thank you, Omar. Dr. Johnston, go ahead.
Umar, I too enjoyed your presentation, and with the understanding that the goal is to understand and get chronic pain under control. My question to you is, what is your most meaningful information that you can extract from this that you see would translate or transition into how we're treating these patients? And my second component is, what's your vision of what changes we're going to need to make?
Okay, those are both great questions, both big picture questions, which I like. And so I think my first answer to the question might address a little bit of both. And so the most meaningful like contribution, or like that thing that I learned in regards to a contribution, is always keeping an open mind in regards to, people talk about that in regards to motivation and things like that, but always keeping an open mind in regards to even like clinical approaches and basic science approaches, because as I was saying earlier, like people have not looked into this as much as they should have. So sometimes, if a patient comes in, whether it's pain, whether it's other processes or other diseases, and it doesn't like fully fit exactly what the picture is, and it doesn't match any of the cases that you go research and look at, always sort of being, always questioning and always seeing what else, what could it be? And like, there has to be a balance between the time and all the other responsibilities that you have. But I learned that like always trying to keep an open mind, always looking out in terms of what could be, trying to learn more and trying to just always further understanding. Now, in regards to the second question was in regards to like what I hope to see in regard from the research. Is that correct? So
yeah, what changes you think we would need to be making?
So, and this is a lot of speculation as well, but like in a sort of ideal scenario, I'd want to see, especially with like, the new therapeutics out in regards to like migraines, I'm sure a lot of us have seen those commercials with the monoclonal antibodies, but my hope is, once there's enough solid evidence that we don't keep putting on Band-Aids, like as opiates, for treatment of pain, because a lot of times, and even and doctors are very well aware of that, where like it's like the patient has pain, but we can't really do much more other than kind of help make it better. And so my hope and my dream is to see something where, whether it's a surgical procedure or whether it's a more targeted medication or other therapies, whether it's like spinal cord stimulation therapy, for example, which is making headway, but is to see us addressing the root cause of the problem. And overall, at the end of the day, just, when patients come in and say, doc, my pain's gone away, whether it's from an epidural or from spinal cord stimulation, it's like it's a very satisfying feeling. Because I did a pain management rotation last month, and it's definitely something that like everyone can relate to, in regards to like everyone's had some sort of pain before, and so everyone can understand like how destructive it can be in life, or how much it just can sort of interfere with daily life. And so that would be my goal, in terms of hoping to see if there's enough research to find a targeted therapeutic for this.
Yeah, absolutely. When it works, it's great and it's very beneficial. When it doesn't, it's frustrating. I couldn't help but notice as you opened the article and you had a list of contributors, I honestly thought that the gentleman on my screen was Michael Burton was also a participant in the authorship, as the co-author. Did I get those names mixed up?
No, Michael Burton, he's actually over here in the presentation, and he's the PI for the lab. And
yeah,
so
that's what I thought. So
yeah,
I wanted to see if Michael had any comments.
Oh yeah, no. This was great. I just was supporting Umar. I've known Umar for a very long time, and it's really great to see him just become the professional, mature person that he is at this point. It's insane.
Yeah.
I met him when he was a baby, but yeah, no. I was just kind of being quiet and just listening, and it was a phenomenal presentation, Umar. So good job. But in terms of, the lab is, so my lab is actually pretty new. I started my lab just a few years ago. When I met Umar, I was a postdoc. And the purpose of the lab is trying to understand, what type of alternations that we can make with therapeutics to help people more efficiently. And so the idea is, does it come down to personalized medicine, which is, a topic we could talk about for ages, but it definitely does change the way we approach pain management and pain medicine, because some of these therapeutics that supposedly only work in X% of people, maybe that's even half of the actual goal. And so if we can discover, well, maybe we need to give a drug that targets this particular kind of cell this way versus this particular kind of cell this way, we change our targets and our therapeutics to the percentage of the current ones to even be better, is because we never, that's another argument, no one ever thinks that opioids are the end all end all be all, but at the same point, that's what we have, and it's kind of the best tool right now. So we want to be sure that if there's a better way that we can utilize them, we should be doing that too, just as well as trying to find alternatives as well.
Sure.
So,
Thank you very much.
yeah, of course.
Dr. Carilla, I saw you put your hand up. You have a question? Go ahead.
I just have a comment. It's my honor to be able to attend this. Umar, I think you did a beautiful job.
Thank you.
And awesome work and a really valuable contribution to the efforts for something that is very important and very far-reaching. So thanks for your work, and thank you, Dr. Burton.
Thank you.
Yeah, I'll say in working with Umar and him showing me some of this research, the one thing that I got from talking to him is the amount of grit and resilience it took him to get through that project. And I think you see Dr. Burton shaking his head, yes, that's a previous mentor of his. And I think some of the mentors in the room now can continue to agree that he has that. And for me, kind of being just a person that's on the outside here, I feel just extremely grateful to think that he's going to be the doctor of the future that's going to have that resilience and that grit to treat me and find out what's wrong with me and that root cause. And so that makes me feel good. So thank you, Umar, for bringing this to us and for showing me those characteristics and hopefully being an example for everyone else on what it takes to really get through a tough project. So, Dr. Wolf, I saw your hand went back up. Go ahead.
Yeah, well, yeah, okay, I am on. I also forgot my first question that I was going to ask, and that was, you mentioned you use the fibers to look at the sensitization and stuff. Did you ever go back and try to correlate the sensitization data with what you actually saw with the macrophage data? Because it seems to me that many times people just start testing stuff and assuming that what they're seeing matters. I mean, classically, you find a gene and, oh my God, this must be the gene that causes this, that, or the other thing. And I'm more of the functionomics person. So, when you get back to, to what really matters in the outcome, are you seeing functional differences in pain responses in these mice that correspond with what you are seeing with your two photon signaling? And would that in any way influence what you might think going forward if you've not done it?
So once again, great question. And so I should have put up the behavior charts, which are kind of, like I said, what we use to go ahead and assure that these are in chronic states. So that's what we started off with first. Like, it wasn't like the imaging or the macrophages, because it was already stuff that we had hypothesized, okay, potentially a difference. But so when we did the surgery, after the testing, like 1, 3, 5 days, 7 days after, we went ahead and did the Van Frey filament testing. And if you looked at the behavior charts, it would be like, you can see like, okay, one day, and then you can see like it like immediately peak up, and they would be at a chronically sensitized state. And if you look at other data from labs that we published and like more long-term projects of other, like just in general consensus, once it sort of spikes up around day 3, it stays at a chronically hypersensitive state where they respond to like the lower filament strength throughout, like a couple days after, couple weeks, couple months after, they're chronically hypersensitized. And so that was a good question. So that's the basis and the foundation that we already have done before plenty of times when we know to be true, that at this spared nerve injury model is like some 10, 20 years old. It's a well-established model that we know behaviorally and clinically, like functionally, we do see, yes, they are sensitized, they are in pain, like they wince, they withdraw their paw. Cold acetone testing as well, where we kind of squirt a little bit of acetone on their leg, same thing. Their withdrawal, they move for that's measured more in time and not withdrawal thresholds in terms of how much they move their paw and have grooming behaviors. And that as well was increased as well. And that's chronically throughout, the whole time. And there's not many.
Were there gender differences? I mean, if you were trying to find gender differences, that was what I was specifically interested in, because if you weren't, then your postulated differences in macrophages may not be giving you the true answer.
So in terms of behavior, and I have to double check on this one because I don't want to give you the wrong information, but overall, from my understanding, they're both relatively sensitized, but the approach that we were taking is to go ahead and in regards to, so there is they do have they both go into chronic pain and they both are hypersensitized. And in regards to gender differences, there are other papers by like Dr. Price's lab that already have looked at other like RNA PCR kind of sequencing data that show that there's different like TLR4 mechanisms that males mainly rely on versus question mark for females kind of thing. And so that's like sort of foundation that we had in regards to that. So the behavioral aspect, more of this, while I understand the functionality component that you're talking about, was mainly to ensure that both were in a chronic hypersensitive chronic pain state. And so that was the main portion of that. And we already knew that there was some difference, there's some difference, but we don't really know in regards to the molecular aspect of this. And sort of, we were just trying to like add to or find out another piece of the puzzle in regards to, and yeah, like you were saying earlier, there could be something else for sure, 100% that might be contributing to these differences. But this is the aspect that we were looking at. But that is a astute.
Thank you. Thank you.
Awesome. Well, thank you again, Umar, for bringing this, and we will put this recording up so you can share it and others can view it. And I thank you guys for coming, everybody. Dr. Burton also for supporting Umar. We always appreciate it. And next week is spring break at KCU, so we don't have anything scheduled, just so you guys know. But we'll be back in following weeks. Keep an eye out for announcements and calendar invites. Everyone have a nice weekend. Enjoy the nice weather.